and we have to learn the structure of two things, that is the centriole and the cilia and flagella. Right. So last class we have drawn the structure and we have studied about the structure of a centriole. Right. And if you remember, the centriole structure will equal the organization is 9 plus 0 and 9 plus 2. Centriole structure to keneke denote kore 9 plus 0 and 9 plus 2.
9 plus 0 organization in centriole.
It is the 9 plus 0 organization that is seen in case of a centriole. After discussion of that structure, we have talked something about a basal body, right? I think you all remember basal body ki hoy. What is the basal body? It is that part from where the cilia or the flagella arises, right? Basal body is nothing but the centriole itself, but provided the position is different. Centriole will come when it is near the nucleus and when the centriole moves to the periphery of the cell, right? And then at that time it is called as a basal body, right? So we have discussed about basal body and centriole, the differences in the form of a chart. Another thing that we have discussed was a little difference between the cilia and flagella as to their number is concerned, their movement is concerned. Exact, we already discussed for it. So, we'll start the class. We are continuing that cytoskeleton itself, but today we'll start the class with the structure of the cilia or flagella. So, heading for structure of cilia, flagella.
Okay, ma'am, aaj lecture 11 Nikki.
Aaj 11 Nikki.
Yes, ma'am.
Aru kundai confirm karo asun. Is it 11 or is it 12?
Ma'am, 11.
11.
It should be 11, right? Okay, okay. Thank you so much. Okay. It should be lecture 11. Okay. Okay, we'll start with the structure, and as we have already discussed in the last class, whenever we are talking about the structure of a centriole or it might be of cilia or flagella, we know it is made up of K. It is made up of certain microtubules, right? So 9 microtubules in the centriole and 9 microtubules in case of cilia or flagella also. So ultimately, K microtubule is one nota. Right? So nota kilwa.
Ma'am, video to freeze me with you.
Yes, ma'am.
Yes, ma'am.
Flagella, what we remember from the last class, that we have to talk about two structures, no? 9 plus 0 and 9 plus 2, right? So 9 plus 0 and 9 plus 2 duta structure or 9 plus 0 structure, that is the centriole. We have discussed it in the last class. Today we are drawing the structure of cilia and flagella. Talking about cilia and flagella, one thing that we already know, obviously the organization would be 9 plus 2, right? Cilia, flagella, or it might be a centriole. It would be made up of what? It would be made up of certain microtubules. What is the number of microtubules? In case of centriole, it was made up of 9 microtubules. In this case also, you will see 9 peripheral microtubules and 2 central microtubules, right? So the structure is something like this. You see 9 peripheral microtubules, and these microtubules will have subtubules. Just compare this diagram with the diagram or the structure of the centriole. In case of centriole also, we have drawn 9 microtubules or 9 peripheral microtubules, right? কিন্তু তাহাঁতৰ কি আছিলে যে তিনটা সাব ইউনিট বা সাবটিউবুল আছিলে কিন্তু in this case we see just 2 subtubules, that is A and B, right? Next, again another comparison is in case of a centriole, this covering or this membrane was absent, but in this case we see a protoplasmic sheet that covers the entire structure. Next, first to kolu subtubular duta, centriole kita asle tinta A, B, and C. Here we have A and B. Protoplasmic sheet present here, absent there. In the center, we see 2 microtubules. Center did not have microtubules in case of centriole, right? Next, we talk about the linker. In case of centriole, CA linker we have discussed. Now we have AB linker. So A pura B locate next right? A microtubular pura dual arm rabo. One extends to B and the other is in the form of a hook. So dual arm rabo. Right. So this is the structure, draw the structure, and here are certain points. We'll discuss the points later. Can you see the diagram properly? Latest dekhisa no?
Yes, ma'am.
Okay. So the points that we have discussed, it is a microtubular structure, the structure of cilia and flagella. It remains bounded by a protoplasmic sheet. The center is called a central exonium, right? And this central exonium is made up of microtubules. Kimanta microtubules, 9 plus 2. You have 9 in the periphery, that is called as peripheral doublet. Why we are using the word doublet? Because we see 2 subtubules here, right? And this 2 is in the center. Center duta ase, but even then they are said to be a central singlet, right? Yate ki koise peripheral doublet. আৰু ইয়াতে কি কৈছে singlet. Why is it called as singlet? Because the center যোনটা microtubule থাকিব তাহাঁতৰ subtubules নাথাকে কিন্তু peripheral যোনটা microtubules থাকিব তাহাঁতৰ subtubules থাকিব দুটা so doublet, right? Next, if you're talking about this peripheral doublet, কি থাকিব microtubule A and microtubule B, and obviously microtubule A would be larger as compared to microtubule B. Microtubule A 13 filaments and microtubule B would be made up of 10 protofilaments. এইখিনিলৈকে লিখা লিখা হলে কবা Is it done?
No, ma'am.
Okay, Nikhil.
মেম
Yeah.
মেম এইটো চেন্ট্ৰেল সিংলেট কিয় বুলি কিয় কয় আকৌ বুজাই দিব
Central singlet কৈছে কারণ central যিটা microtubule আছে তার subtubule নাই, right? If you see it in the center, ইয়াতে কেইটা microtubule আছে দুডাল, right? So this green structure, these are two microtubules, but they do not have a subtubule. সেইকাৰণে singlet বুলি কৈছে। কিন্তু if you are talking about this peripheral গিটা তাহাঁতৰ প্ৰত্যেকটোৰে কি আছে দুটা দুটা subtubule। তাৰমানে this is one microtubule with two subtubule. This is one without a subtubule. This is another one without a subtubule. Clear?
ওকে মেম য়েছ মেম
First দুটা point হৈছে নেকি? Point 1 and point 2?
Yes, ma'am.
Yes, ma'am.
এইটো point হৈছে central axoneme.
Yes, ma'am.
য়েছ মেম
Okay. So subtubule A কি থাকে দুডাল arm থাকে. I think it's clear in the diagram. One arm ends in a hook. Hook like structure এটা থাকে, and the other arm attached with microtubule B. Next. Next point, The central pair of pupils are connected to each other by a double bridge and is covered. তাহাঁতৰ এখন membrane থাকিব covered by a protoplasmic sheath, and this sheath is called as central sheath. Right? So this is what is the central sheath. Point number 4 complete.
Yes, ma'am.
Is it done? Point number 4, can I rub it?
Yes, ma'am. Yes, ma'am.
So from this section, I think I have already said in the first class that protein guitar num more about questions. What is the protein? Right? Or cilia or flagella is made up of which protein? So TB protein you need to remember. Next we have to talk about 2 more proteins. Is point number 5 over?
Yes, ma'am.
So protein kita question you have to read tubulin, dyamine, and nexane. Right? Next time question has been asked about this one. Arms of A, which protein is present? Right? Another point about the doublets, and when I write doublets, we are talking about this peripheral microtubules. The doublets are tilted at an angle of 10 degrees. If you remember, we have talked about such a statement when we were discussing about the structure of the centriole. In case of centriole, what we have seen is tubules থাকিব কেইটা tubules 9টা, and they will have three subunits, right, or subtubules, and these subtubules would always be tilted and they form an angle with the center. আৰু কোনটো angle বা তাহাঁতৰ যোনটো tilted আছিলে ন, tilted angle, what was the angle? You don't remember. ইয়াতে আমি কি লিখিছো? They are tilted by 10 degree. তাত কি আছিলে?
40 degree.
45 degree.
45 degree. Very good. Very good. So this is how you compare both the structures and then you study. It's not like this, since from MCQ point of view, obviously structure to delineate and you are not asked to identify, right? So the questions that you need to form yourselves are 9+0 car, 9+2 car, protoplasmic sheet বা covering car আছে, car নাই, right? Next is the types of tubules. Next is subtubules কেইটা আছে, দুটা কাৰ আছে, তিনটা কাৰ আছে. Next is we have to talk about the proteins. So এইখিনি আছিলে the structure of the cilia and flagella. Is the structure clear? এইখিনি points খুবই important. Next, functions, and whenever we talk about cilia and flagella, the first function that we remember, they are the organs of locomotion. Cilia থাকিব, flagella থাকিব. Another point is one second. So in short, functions বুলি ক'লে these two functions we remember. Functions of the ইমান questions আহা নাই. So this is all about the cytoskeleton, right? From the starting itself of the chapter, what we have studied this, we have talked about the two different types of cells, no? Prokaryotic cell, eukaryotic cell, plant cell, animal cell. Like discuss korilu, and then we were discussing about the contents of a cell. Talking about the contents, cell wall, cell membrane, these are the things that we have discussed. The different types of organelles, right? Some of the organelles that comprise the endomembrane system. The other organelles that do not belong to the endomembrane system. right? And ultimately, what we have studied is the cytoskeleton. After this, what we need to talk about is something called as microbodies, right? In the cell, যিমান major organelles আছিলে, এইবিলাক organelles we have already studied about it. But apart from these organelles, no, কিছুমান সৰু vesicle-like organelles or small sac-like organelles are also present, and these small spherical structures are something that is called as microbodies. So এটা cellot যে ডাঙৰ organelles বিলাকে থাকিব, not necessary, right? Small vesicle-like, vesicle বুলি ক'লে I think you remember vesicles, no? We have studied in Golgi bodies and endoplasmic reticulum. Vesicles কি হয়? Small sac, spherical sac-like structure. These are called as vesicles. No? So তেনেকুৱা ধৰণৰ সৰু structures কিছুমানো থাকে. And these are called as microbodies, and we have talked about 3 different types of microbodies that are present in a cell. So if this is over, next heading কৰিবা microbodies. এইটো হল নেকি?
Yes,
Yes.
ma'am.
Okay. Heading for microbodies. Right. So there are certain small organelles which are called as microbodies. Small single membrane bound organelles that are responsible, or inside these organelles বহুত ধৰণৰ কিছুমান enzymes থাকিব, right? And these would be responsible for different types of reactions. So বেলেগ microbodies are বেলেগ functions হ'ব. So we'll be talking about, one second. Three types: stereosomes, peroxisome, আৰু spherosome, peroxisome, আৰু glyoxysome. And while we were discussing this chapter, if you remember, we have talked about this word, eta statement কৈছিলো. If anyone who remembers, spherosomes act as dash in case of a plant, or in case of a plant cell. Spherosomes act as dash in case of a plant cell. Or spherosomes are also called as dash. Understand? Something related with lysosomes. Remember the organelle called as lysosome? আমি তো সেইটো পঢ়িছো ন? Yes or no?
Yes, ma'am.
Okay. In case of lysosome, lysosome is a characteristic of a plant cell, ne animal cell. Ne, it is present in both. Ne পাহৰি গলো.
Both.
No, in case of an animal cell, right? In case of a plant cell, spherosomes are considered as plant lysosomes, right? Hetu statement he ami porisilu. Eti ami aru okman details pohim, right? The statement that we have studied is plant lysosomes are called as spherosomes, or spherosomes are called as plant lysosomes. Okay, that is one statement that we have studied. What about peroxisome? Peroxisome apote porisa? Have you heard about this before?
Yes.
Yes, ma'am.
Right? Peroxisome
ma'am.
ami kihot porisilu?
C2 cycle.
C2 cycle, or it was also called as photorespiration, right? So in case of C2 cycle, তার আমি কি দেখিছিলো যে peroxisome কি হয়েছিল না? Okay, we'll be writing about it. So if this is over, spherosomes heading করিবা. another thing, before writing about these microbodies, no, ইয়াৰ পৰা question কি আহে, microbodies কেইটামান points মইতো mark কৰি দিমেই, তাৰ উপৰি who has discovered the respective microbodies সেইটোৰ ওপৰতো question আহে, so scientist জনৰ নামটো ভালকে পঢ়িবা. এইখিনি বেছি বুজাব লগা একো নাই. Abundantly found in endosperm cells of oil seeds. So discovered that Turner half membrane bound organelle বুলি কিয় কৈছে, because they are single membrane, right? They are spherical. Main function is storage of fats and lipids. They are found only in plant cell, abundantly found in endosperms of oil seeds, right? And next is they contain hydrolytic enzymes. They contain hydrolytic enzymes and are believed to be called as plant lysosomes, and they arise from small endoplasmic, smooth endoplasmic reticulum, right? First of all, what I said is these are nothing but small vesicles, no? Vesicles, obviously, they are formed from endoplasmic reticulum and Golgi bodies. So this is the information about spherosomes. Now diagram and anything, these are also not important. So in a cell, apart from the main organelles, we see certain vesicular or certain small spherical organelles that are called the macrobodies. One of them is the spherosomes. Discover kune korisil, arise cobra hoisil, considered as what? They are considered as plant lysosomes. If this is done, peroxisome heading koriba.
Ma'am.
Ma'am, স্পেৰোজোমছ বিলাকেও সুইসাইড বেগছ হিচাপে কাম কৰে নেকি?
Exactly তেনেকে নহয়, কিন্তু যিহেতু এনজাইমছ বিলাক আছে ন, আৰু কোনোবাইটো ক্লিনিং মেকানিজম কৰিব লাগিব even in case of plant. So they are considered. Proper definition এটা তুমি দিব নোৱাৰা. You cannot be saying it spherosomes as the suicidal bag of a cell. এই wordটো অকল lysosomes হতে থাকে। কিন্তু যেহেতু hydrolytic enzymes থাকে, enzymes বিলাকে cleaning mechanism কৰে, they are believed to be plant lysosomes.
Okay, ma'am.
আৰু যোনকেইটা point ঠিক কৰিছো এইকেইটা ভালকে পঢ়িবা। Please tell me if it is done. Is it over?
Yes, ma'am.
Okay. Next लिखा peroxisome. Again, discovered by Duve, D-U-V-E.
Ma'am, discovered by Ma'am D. Duve.
Duve, ha. Okay. Peroxisome কিয় আছে? কাৰণ তাত hydrogen peroxide পায়, right? In this small site-like structure, you see the presence of certain types of enzymes, and these enzymes are responsible for producing the peroxide and also destroying the peroxide, right? So, এটা stackতে বা একেটা structure-তে peroxide produce কৰা enzymes আছে আৰু সেই যোনটো produced হৈ গ'ল সেই peroxide টো ভাঙিবলৈও তাহাঁতৰ enzymes আছে. And we know whenever the peroxide gets broken down, কি form কৰে? H2O2 যদি ভাঙি যায় কি ফৰ্ম কৰিব? কি ফৰ্ম কৰিব? H আছে, O আছে, obviously water ফৰ্ম কৰিবই, right? Okay. তাৰ ভিতৰত discovered form important, arises form important, আৰু কি enzymes আছে? Some of the enzymes that you find. এইখিনি হল এক্সট্ৰা, এইখিনি পোৰ্শন ইমান ইম্পৰ্টেন্ট নহয়। এনজাইমছৰ যিখিনি নাম, ইমান নামৰ ওপৰত কোৱেশ্চন অহা নাই। Next, the peroxide destroying enzymes, that is catalase বিলাকে কি কৰিব? And finally, the last point. They perform photorespiration. Oh, green cells are animal cell or likha nahi no, likha in animal cell. Detoxification
Ma'am,
of alcohol.
peroxide destroying enzymes to catalase লিখিছিল আপুনি।
Catalase, catalase,
Why নাই no?
নাই.
Okay, ma'am.
green cells to thakibo ba plant cells to thakibo animal cells to thakibo. So apart from producing water and oxygen, in animal cells detoxification not help kore. This is the extra point okol etu lagibo kintu. Photorespiration buli kole I think you remember no C2 cycle, also called as ki C2 buli kyo koisile janane? Two carbon compound that is glyoxylate. Is it over?
Yes, ma'am.
Next microbody is glyoxysome.
मैम लास्ट पॉइंट तो हुआ ना
Animal cells or eto.
यस मैम
Okay. Next is glyoxysomes. Last to likhi lo.
मैम हो
Next glyoxylome.
মেম এইটো গ্লুকো কি লিখা আছে প্ৰচেছ
Gluconeogenesis. GLUCONEO. Gluconeogenesis. GENESIS. Last line to etu nalage. Yate glyoxylate cycle likhisa no. A glyoxylate cycle to side ote likhason. Absent in case of animals. Right? So glyoxysome, so fully we understand we are talking about a body that contains certain types of enzymes. A enzymes kini, junkini glyoxysome of ase. What is the function of this enzyme? The function is these enzymes will convert the fats into carbohydrates. So central bitor jikini fats thake no, he fats kini ki hole convert koribo carbohydrates hole. And this is possible only in case of plants and obviously not in case of animals. Think about a condition যখন আমি খোৱা বস্তু খাই আছো সেলত ফ্যাটস আছে আর ফ্যাটস বিলাক আকৌ ফুডলে কনভার্ট হৈ গৈছে তেতিয়াতো আমাৰ প্ৰবলেমে সলভ হয় না নহয় বেসিক্যালি তো খাবও নালাগে second there is no point of or there is no tension of getting what fat জিম টিম যাব দৰকাৰ নাই আৰু সেলৰ ভিতৰতে হৈ যাব right so animal cells of fats কেও carbohydrates হলে back convert নকৰে in the case of plants fatsবিলাক কি হ'ব converted back to carbohydrates and this cycle is called a glyoxylate cycle. Is it clear? So
Yes, ma'am.
18 about the 3 different types of microbodies seen in a cell. If it is over, please say.
Yes, ma'am.
Last line.
Okay, right.
Done, ma'am.
Okay. So we are almost at the last portion of the lesson. Our last portion of the lesson, what we need to talk about is duta major thing. One is the nucleus, second is the chromosomes, right? Nucleus or chromosomes, one discussion koribo লাগিব. So we'll start the nucleus. Okay, talking about the nucleus, what we know, the nucleus ki hoy? First point, discovered by. It is already পঢ়ি আহিছো, discovered by কোন? Who discovered the nucleus? নে পাহৰি গলো?
Robert Hooke.
Robert Hooke নে Robert Brown?
Robert Brown.
Robert Brown.
Robert Brown, exactly, not Robert Hooke, right? So discovered, Kune discovered korisile. Next is talking about the nucleus. Obviously, it is a master cell organelle right? যেনে কিছুমান tag আছে no kitchen of a cell of যেনে chloroplast বুলি কয়, master cell organelle বুলি কলে we know that it is the nucleus, right? Suicidal bag it's a lysosome, etc. Next এটা nucleus ৰ ভিতৰত কি থাকে? Genetic material থাকে, and then someone who has first proved it, এতিয়া আমি জানো যে তাৰ ভিতৰত genetic material থাকে, right? কিন্তু সেই বস্তুটো কোনে experiment কৰি পেলাই ওলাইছিলে, even the name of the scientist, that is also important. So these are the things that we are going to study here, and together with that obviously the structure of the nucleus. Talking about the nucleus, which is made up of what? অকমান কি rough idea আমাৰ আছে. We know that the nucleus is a double membrane structure. Inside it genetic material in the form of certain thread-like structure that is called as কি? Chromatin. Chromatin বিলাক কি হয়? Chromosomes হলে condensed হৈ যায়। কেতিয়া হয়? During the process of cell division. These are the things that we know, so we'll be talking about all this. So first, nucleus heading করে introduction অকমান লিখিম। Together in this unit, we have to talk about the number of nuclei per cell also, right? According to cell theory, কি কৈছিলে? যে each and every cell will have one nucleus, but we have seen certain exceptions. We have seen cells that do not have a nucleus. We have seen cells that have a multinucleated condition also. Next, we have to talk about the position of the nucleus in a cell. Normally, cell will be a plant cell, animal cell. In a broader sense, we discussed. So, in case of a plant cell, position of the nucleus key, or in case of an animal cell, position of a nucleus key. So, these are the introduction or introductory points. এখানে আমি লিখিম। কিছুমান points লিখি লওঁ। So, A point to important, ঠিক আছে? এটা তো important কোনে discover কৰিছিলে। Next, we have to talk about, whenever we talk about nucleus, we know কি থাকে? Chromosomes and obviously responsible for কি? Hereditary, right? Transfer. Next, এটা নিউক্লিয়াছত যে হেৰিডিটাৰী মেটেৰিয়েল থাকে কোনোবাইটো প্ৰুভ কৰিব লাগিব। So it was an experiment of J. Hammerling. Question আহে J. Hammerlingৰ experiment যে what did it prove? It proved that nucleus contains hereditary material. The second point is Hammerling কিহৰ ওপৰত experiment কৰিছিল? He has conducted the experiment on a green algae, and the name of the algae is Acetabularia. Have you heard about Acetabularia before?
Yes, ma'am.
It is also called as কি? আমি algae পঢ়োতে কৰিছিলো ন? Acetabularia. No. Acetabularia. Acetabularia. Why we need to talk about this specific green algae? Because it is supposed to be এটা কিবা এটা unique point আছে কি আছিলে? First, it was also called as, yeah, very good. Say, please say.
Largest green algae.
Largest green algae নহয়, largest unicellular algae. Right? So it is the largest unicellular algae. So that point makes it unique. So Acetabularia, that's the first point. Second point, it is also called as the umbrella plant. right? I think you'll remember when we have talked about algae in plant kingdom, right? Green algae, brown algae, and red algae. Green algae, we have given certain examples of unicellular green algae, multicellular colonial green algae, etc., etc. Unicellular green algae, eta ki asile? Chlamydomonas. Remember, another was chlorella. Another example was Acetobularia. Right? So Acetobularia, I'm more rakhibo. Next point is, we have to talk about number of nucleus in a cell. When we talk about number, we talk about uninucleate condition. Sorry. Uninucleate sobote thake maximum. So hetu likhibo nalage. Binucleate in case of Paramecium, multinucleate in case of any examples like Vaucheria, Rhizopus, Elbugo, etc. Right? As far as multinucleate is concerned, mainly confined or mainly seen in which group? Obviously fungi. So Rhizopus, Elbugo, nothing but examples of fungi. Talking about Vaucheria, what is Vaucheria? It is nothing but an algae. Algae, algae, we are talking about green algae, red algae, and brown algae. But apart from that, we have something called as yellow-brown algae also that belongs to the Gentophyceae, right? There is Rhodophyceae, Phaeophyceae, Cyanophyceae. We have talked about. No, sorry, Rhodophyceae, Phaeophyceae, or Rhodophyceae. So, that is called as Xanthophyceae. Xanthophyceae, as an example, hold Vaucheria. Iman nalage, what do you remember? That this is an algae. Right? Next. So talking about nucleus, according to the cell theory, one nucleus per cell, but we see multinucleate and binucleate condition. Examples we have discussed. Next, whenever we are talking about multinucleate condition, we have to talk about two terms. No, do you remember the term that was given to multinucleate condition in a plant cell and multinucleate condition in an animal cell? Ki boli koi? Eta cellot jodi bahutta nucleus ase, multinucleate to it's understood, but we had specific terms for that. Key condition?
Syncytium.
Syncytium and Coenocyte, right?
Yes, ma'am. Syncytium or Syncytium in case of animals, and in case of plants, we use the word called as Coenocyte. Next, talking about position of the nucleus.
Ma'am, picture blur hoise, ma'am.
Picture blur hoise. So, bore hoise na apun tomar hoise.
Yes, ma'am.
Yes, ma'am, hoise.
It's blurred. Completely blurred. You cannot see anything.
ठीक हो गया
ठीक हो
Cool.
गया
Okay. So next is number of nucleus we have discussed per cell. Next is position of the nucleus in a cell. We use the word called as centric. Centric nucleus or central nucleus is a characteristic of animal cell. Eccentric is another word that we use. That is a characteristic of
মেম
হা
মেম অকণ সময় কি কৰালে মোৰ অলপ কাট হৈ গৈছিলে
এইখিনি পোৰ্শন লিখিছা নাই নাম্বাৰ অফ নিউক্লিয়াছটো
মেম এলবুগলি লিখিছিলো
এলবুগলে লিখিছা ন তাৰপিছত আৰু একো কৰাই নাই মই খালি এইখিনি পাৰ্ট কৈছো যে whenever we use the word multinucleate we understand one cell having more than two nuclei that is called as a multinucleate but we have certain terms in case of a plant cell and in case of a animal cell সেইটাই লিখিছো যদি multinucleated condition in case of a animal cell হয় then we use the word called as syncytium and if it is a multinucleated condition in case of a plant we use the word sinucyte সেই দুটা word লিখিছো
ওকে মেম
Okay. আৰু ভাউচেৰিয়াটো লিখিছা নেকি এলগা
ইয়েছ মেম
ওকে So we have seen that if you're talking about the position of the nucleus, it's not like this, that it will always be in the center. So the center is centered, and similarly, in case of a plant cell, away from the center, so we have used the word called as eccentric. Ketyaba plant cell, or instead of eccentric, we can also use the word called as peripheral. Because the side of the we can use the word peripheral. Oh. Adipocytes. Adipose tissues spelling ভুল হৈছে Another term, interphase nucleus. Interphase nucleus কি হয়? The nucleus of undividing cells. That is called as interphase nucleus. Next one is about the shape of the nucleus. If we talk about the shape, it would be ovate or oval. G likha oval. Oval in case of smooth muscles, bilobed in case of acidiophils, and trilobed in case of basophils. Okay, smooth muscles we understand. What is acidiophils? Acidophils কি বস্তু? Have you heard this term before? Acidophils?
আৰ্কিবেক্টেৰিয়াৰ এটা টাইপ হয়
Very good. কি আছিলে ফুলটো? আৰ্কিবেক্টেৰিয়াৰ এটা টাইপ, নামটো কি আছিলে? Thermoacidophils, right?
Yes, ma'am.
So thermoacidophils or acidophils, we are talking about acidophils. What does it indicate? It indicates those organisms that can survive in acidic conditions, right? আমি thermoacidophils archibacteria পঢ়িছো. So acidic conditions of যোন থাকিব পাৰে acidophils. Now what is a basophil? কি হয়? Acidophils পঢ়িছো, so that those can survive in acidic condition. আৰু basophils মানে কি? কি হয়?
হোৱাইট ব্লাড চেলৰ এটা টাইপ
Yeah, it is a white blood cell or white blood cell বুলিয়ে কৈ থব পাৰে. Now you don't have to understand to say that যোনবিলাক basic or base of থাকি ভাল পায়, those are called as basophils. তেনেকা নহয়, ঠিক আছে? So basophils বুলি ক'লে if you are confused, white blood cells বুলি লিখি থৈ দিব পাৰা. Right? So today up till here what we have done is we have talked about the microbodies. Obviously we have talked about microbodies. We have completed the cytoskeleton and nucleus আমি আৰম্ভ কৰিছো. This is the introduction of nucleus, some of the introductory points, and after this we have to talk about the structure of the nucleus, right? So এইখিনি লিখি লোৱা. Talking about the structure of the nucleus, I don't think we'll be able to complete it in today's class. So this is something that we will do it in the next class. Okay? এইখিনিলৈকে কিবা doubts আছিল নেকি? Okay, if suppose you don't have any doubt, I think we can wind up the class today. We'll meet you in the next class. লিখি
Ma'am,
থৈ
last
দিবা
point
the
লিখা নাই
structure of the nucleus. Please write.
দিব নেকি?